Okay, guys, this is chapter 23, immunity, and this is going to overlap completely with microbiology. So if you've had microbiology or if you are taking it currently, this is probably very, very familiar to you. So good, good for you. If not, you're going to get this again in micro, so it's a good overview. All right, so let's take a look at our first picture here. So our immune system, our immune system is sometimes thought of having three lines of defense. The first line of defense is going to be just physical barriers. These are things that you're born with. They're just going to block the entrance of microbes into your body. So under letter A, we have physical barriers, which is your skin. So your skin is a really good line of defense. It is tough. Here, let me show you a picture, right? It's dry, it's tough, it's salty, it's thick, it's, not very hospitable to microbes. Of course, there are microbes that live on our skin, yeast being one of them, but it's sort of an environment that is difficult for microbes to penetrate. Okay, so intact skin is really great. And then we have our second, so skin is on our body, of course, everywhere that's not mucous membrane. Mucous membrane is a second layer that is going to be very good, and it traps microbes. So our mucous membranes, of course, like our mouth or our eyes have the conjunctiva. They're made to create materials like mucus or tears that would, could trap the microbes and then move them out of our body. So the mucous membranes is also really great. And then we have some areas that have cilia, right? So our respiratory tract has cilia to help sweep those substances up and out of our lungs. And our gastrointestinal tract also has mechanisms like a lot of mucus, a lot of goblet cells to trap those microbes. So those are some physical barriers. And then let me just show you a picture of our nasal mucosa, right? So the cilia here is going to be covered in mucus, and then so if a particulate that you're breathing in through your nose will get trapped in that mucus, and then you could blow your nose, the mucus kind of just goes, you could swallow it and sort of gets rid of the microbes that way. In terms of chemical barriers that we all have, chemical barriers would be things like sebum. So on the skin again, if we want to look at this one. So sebum, created by our sebaceous glands, is our oil. And oil, by its nature, right, are fatty acids. So the acidic portion of that fatty acids actually inhibits some microbes from living on our skin. And then, of course, we have acids in our stomach. So things we take in through the food can be killed in the acids of our stomach. And then there's also other fluids. There's lysozyme in our body. We talked about this. So lysozyme is an enzyme that breaks down peptidoglycan cell walls of bacteria. It's in high concentrations in the saliva. It's also in really high concentrations in your tears. And then it's in other, smaller concentrations throughout the body, but a lot of your body secretions have lysozyme in it. And then we have acids that are in our urinary tract. And the vagina is acidic. And that acid, the acids again inhibit microbes from taking over, taking hold. Now, this is to say your whole body really has its own microbiota, right? So we talk about let's put the word here, microbiome. There are some areas of your body that remain sterile, and there's other areas of your body that are just inhabited by microbes. And those normal microbes that live on your body are called your microbiome. They're sometimes also called your normal flora. Okay. And for the most part, this is just F. They're fine. They coexist with us. They are, have a symbiotic relationship with our bodies. Sometimes they do cause disease if they become put in a place where they're not supposed to be, or if you have a cut and they go from living on your skin to deeper in your skin, your dermis, then they do cause infection. But for the most part, they're good for us. They keep pathogens away from our body because they kind of set up a turf, right? They have their space, they have their territory, and then they inhibit other pathogens from living or growing on your skin, from taking hold. So those are some of the first defenses, right? Physical barriers like your skin, physical barriers like your mucous membrane, and then chemical barriers that we secrete. And everybody's going to have these, right? So this is called your innate innate immunity, as well as your white blood cells that we're going to get to. So once those microbes penetrate into your tissues, what's your second line of defense? So your second line of defense would be your leukocytes, right? So letter C, we have lots of leukocytes, neutrophils. We looked at these in the blood chapter, so we don't need to go back over them. But we have our basophils, our eosinophils, our macrophages in our tissues, monocytes in the blood, neutrophils. Then we have other cells like mast cells and dendritic cells. Here's a dendritic cell, and I don't see the mast cell in this picture. So no mast cell illustrated here, but they are going to be found in areas that are also exposed to the outside world. So skin and mucous membrane or mucosa. And mast cells, you know, if you look any of these cells up, they have tons and tons of jobs. So they're going to help, the mast cells are going to help other arms of your immune system. They do a big role in secreting histamine. So they do have a role in allergy with that histamine. And then dendritic cells are found also in your skin and mucous membranes, and they're very phagocytic. So they're very similar to macrophages, and they're very phagocytic. And you probably remember Langerhans cells in your skin. That is a form of dendritic cell. And they're called dendritic cells because of these projections that they tend to have that look like dendrites, like the dendrites on neurons. They're not neurons, but they have the same kind of feature to them. All right, so we have these non-specific cells. What does this mean to be non-specific? Right, non-specific. And that means, and this is review, that they're going to respond to any foreign entity that comes in, and regardless of what it is, it's just something that is non-self, right? Something that's foreign. And they are able to phagocytose those pathogens and clear them. So that's like one arm of our immune system. They don't have memory, right? Non-specific, that means that there's no memory. So they don't have an altered immune response. They have the same basic playbook no matter what. So if they encounter a pathogen, they go through the same motion over and over and over again. There's no increased response or decreased response. They just have that same response. Now, your specific cells, your T cells and B cells, right? This is your B cells, T cells. They are considered specific immunity. This is because they respond only to specific antigens that enter your body. So they're choosier as to what creates this immune reaction. However, they change their behavior the second time that they see that antigen because they do create memory cells. So memory cells are made after the first encounter. And so memory is very important. And that's called your adaptive immunity, which we'll talk about in just a second. Now there's a third cell here called a natural killer cell. So natural killer cells, your textbook also calls them null cells, but that's sort of an old name. So I always refer to them as NK or natural killer cells. And they're really important in mediating virus infections. And so they're the first line of defense with viral infections. And they do kind of skirt the line between specific and non-specific because they can respond specifically to specific antigens, but they also have a role in just destroying any cell that's virally infected. So they have like a dual category there. So, but very fast acting and very important for viral infections. Okay, so just that overview of non-specific and specific immunity. So that was kind of that overview. So let's take a look at this picture. So the first line of defense, all right, so this is called innate immunity. Non-specific is also called innate immunity because everybody's born with this. If you have a working immune system, everybody has the same innate immunity. So we're all going to have our first line of defenses. We're all going to have our barriers and our secretions. We're all going to have our leukocytes that behave in a very specific manner. And then we have all these responses, these non-specific responses that we'll look at in just a second. So we all create the same antimicrobial proteins, such as complement. We have the same inflammatory response and fever. You know, everybody gets fever. So there's these set innate responses that we have. And then, so this is our second line of defense, right? So first line, keep them out. Second line, they got in, but we have our sort of universal defense mechanisms. third line of immunity is now we get our specific cells involved. We have our lymphocytes, our T cells and B cells. They create antibodies and they create memory cells. So this is what's called adaptive immunity, because this is different from person to person. So if, a person, it depends on where you live, what you're exposed to, and this could include like, what are you eating, or do you have pets? Do you live in a city? Do you live in the countryside? It's very specific, or it adapts to your environment. So your immune system learns what is around you by basically coming into your body and then, alerting these T cells and B cells and then creating memory. So you get protected from your local environment. All those germs, pathogens in the local environment, you have this adaptation to. You have an adaptive immune response. This is why that people who live in Mexico or in Indonesia can drink the water, right? They're perfectly adapted to their local environment, and they've gotten this immune system to where the microbes that are in their water sources don't bother them. But a traveler, right, if you or I went to Mexico or went to Indonesia and we drank the water there, we might get diarrhea because our immune systems haven't been around that particular microbe, and so we get sick. Okay, so that's just a quick innate versus adaptive immunity. All right, so let's keep on going with our non-specific sort of basic immunity, innate immunity, lymphoid tissue, right? Everybody has lymphoid tissue. And we're not going to go into too much detail because you have the lymphatic system, but we know that we have our bone marrow that produces all of our blood cells, which is, the term is hematopoiesis. Our T cells, right? So this is bone marrow is where hematopoiesis occurs. Then the T cells in particular will travel up to the thymus, right? And they're named T cell for thymus, and there's mature there. And then your peripheral lymphoid tissues, right? Lymph nodes, the spleen, Peyer's patches in the small intestine, other areas called MALT, right? Mucosa-associated lymphoid tissue. Your tonsils, those are all areas of a collection of your lymphocytes as well as other immune cells that survey the fluids, right? So the fluid in your lymphatic tissue, again, is called lymph, right? And so that's the interstitial fluid that moved into the lymphatic system. It's getting screened by those lymph nodes. Okay, so you guys had that. We don't need to talk more about that from anatomy. Okay, so the one difference between the spleen, let me just mention this, is that the spleen does not filter lymph. The spleen filters blood, and that's mentioned in the number 2, the peripheral lymphoid tissues. So the spleen actually gets blood coming into it, and it filters the blood, whereas lymph nodes, Peyer's patches, just lymph nodes, right? Lymph nodes get lymph moving through them. And then these Peyer's patches in the small intestine, right? That's just whatever comes through the intestinal wall. And then your tonsils, those are in your oral cavity, in the nasal cavity. Those are going to protect you against those inhaled particulates, right? Inhaling or ingesting the food, you have those tonsils there for immunity. All right, moving into different pathogens. All right, so if you're not familiar with these different kinds of pathogens, this is just a quick introduction. So the smallest pathogen that we know of that we can mount an immune response to are viruses. And viruses have different sizes, but they're all tiny. Okay, so we can see that let me just say that this guy is an exception. That bacteriophage is only a virus that infects bacteria, so it doesn't concern us. This does not affect humans. We have HIV, we have Zika virus, we have parvovirus. So those are all viruses that infect us. Compare that to a bacteria. Look at this guy, right? So viruses are tiny compared to a bacterium. In terms of, the size differences, there are large viruses, but they're called giant viruses, but these are not human pathogens. So we don't need to worry about them. Although Mimi virus is a cute name. It's a giant virus. And then I just want to introduce, what is a virus? A virus is very interesting because you could argue that a virus is alive, or you could argue the virus is not alive. And so I like to use the term, I heard a professor say, borrowed life. Viruses they borrow life. What that means is that outside of a cell, they are inert. They are consisting of some kind of nucleic acid. This could be DNA, or it can be RNA. The SARS-CoV-2, right, that's an RNA virus. Then that protective nucleic acid, whether it's DNA or RNA, is inside of a protein coat called a capsid. And then sometimes, I'm going to put a parentheses around envelopes because not all viruses have an envelope, but some of them do, which is a phospholipid envelope. And in order for viruses to reproduce, they have to infect a host. They have to go inside a host cell, and then they use the host cell's machinery. They use the enzymes, they use the ATP, they use all of that. They steal it from the host, and they build new viral particles, and then they come out of the virus. So in order for viruses to get in, the proteins that they have on their outermost part, whether it's the envelope, they can have protein, they're called spikes on the envelope, or they could be proteins on the capsid, depending on the virus, but that has to fit a particular receptor on the host cell. So it really fools your cell into coming in. So that's it with viruses. So very small, measured in nanometers, tiny thing. Then the others, right, the ones that we're maybe more familiar with are bacteria. Bacteria are much bigger than viruses. They are clearly cells. They have their own organelles. They have their own metabolism. They can reproduce on their own. And bacteria, remember their cell wall is peptidoglycan, and that is important because lysozyme attacks that protein. That makes their cell wall up, and then we can also make antibiotics to that protein. Actually, it's not a protein. It's basically carbohydrate that has protein linkages in it. But anyways, so that's your bacteria. And then other things, fungi, of course, can come and give us fungal infections. Fungal infections, by the way, are called mycoses, mycoses. And the MYC is what they use as a prefix for fungi. So if you study fungi, you're a mycologist. And there's different forms that can infect you. You know, yeast infections or molds can grow in your body. And one characteristic of fungal infections is that they do grow very slowly in the human body. And since they grow slowly, they tend to die or take a while to die. So treatment usually lasts a very long time with fungal infections. Fungal infections are very frustrating because they do last, months at a time. If you've ever had a fungal infection, like a toenail, sometimes you get a fungal infection in your nails. It just takes a very, very long time to treat. And then there's other parasites, right? So parasites is a big word, and these include things like worms, tapeworms, hookworms, and they also include things called protists, which are going to be amoebas or something called plasmodium that causes malaria. So we're not going to go into any detail on that, but parasites can be both single-celled, small, microscopic things like amoeba or worms. Now, worms can be big, and you can see worms, but we don't get infected by the actual big worm. Most of the time we get infected by their eggs or the larvae that are hidden in our water and food, and then we accidentally ingest them. So I'm going to do a quick, I'm going to go to this website just so you guys can see this. This is a fun one. And so we're just going to go in on this nail here, and you can see that we have a scale. So we have a hair, and then we can see we have some dust mites. And then we are coming into a little collection of particles. And then here we have ragweed pollen. The green guy is ragweed pollen. We have a lymphocyte. We have some red blood cells. We have the baker's yeast. And then we have some E. coli. And we have right beside the E. coli, so that's bacteria. We have some staphylococcus bacteria. And then lying next to that, we can see some other interesting things. That candy cane shaped virus is Ebola virus. Ebola is actually quite a large virus when you look at all the viruses. And then next to Ebola virus are some tiny little particles, and that's the rhinovirus, which is a virus that causes the common cold. There's many viruses, I think 50 viruses that cause the cold. This is one of them. So here we are at 1,000,000 times magnified on the head of a pin, right? So we're looking at basically something around 20 nanometers in diameter. We're going to come back out. Nice, right? So imagine how many rhinoviruses can be on the head of a pin. A lot. All right, let's keep going with our... All right, okay. Now we're moving into our body's defenses, right? So what kind of defenses do we have? Our non-specific defenses are going to begin with inflammation. So you have some kind of injury, right? It could be sort of physical injury, or it could be a chemical injury, it could be something that you have an infection, and inflammation is always right there, the first kind of event that occurs. So the reason we have inflammation is to draw our immune cells towards the site of injury. So we're going to, for letter A, right, we have the first phagocytosis. So those nearby macrophages, the ones that are in the skin, the ones that are in the mucous membrane, right, they're going to be recruited by the chemicals of the microbes. They're also going to be recruited by chemicals that damaged tissue is sending off. And so they're going to be there, and they're going to undergo, they're going to phagocytose those microbes. Then we're going to have a lot of cytokines. So cytokines, that word is a generic sort of general word for all these chemicals that are secreted by immune cells and also by your body cells. And they work on the immune system to trigger different processes, whether it's, recruiting other white blood cells or activating other white blood cells. But that's the word cytokine. So cytokines are going to cause this dilation, and histamine being one of these. So histamine is a very powerful vasodilator. It's also a very powerful vascular, sort of makes these blood vessels very permeable. So what that does is the histamine is going to draw out a lot of fluids from the blood vessels nearby. It's also going to cause the proteins to come out a little bit easier because the blood is more permeable, right? The blood vessels are more permeable. So we're going to have a lot of fluid, and we're going to have a lot of proteins coming into the site of injury, and that's going to create those classic signs of swelling, which is redness. So we have redness from the blood, right? That the blood, we're extruding into the area, or we're dilating those blood vessels so the area gets more red. We have heat also from the blood. We have edema or swelling, obviously from the fluid buildup. And then we're going to have pain because we're compressing those nearby nerve endings, right? And those are all sort of the signs of inflammation. Then we have sort of containment. So we're going to have another active chemical here, heparin. So heparin is an anticoagulant, and your body is going to want to start clotting the tissue, and that could actually wall off the proteins and the cells that are coming out from the bloodstream. So you're going to have heparin that kind of prevents the clotting for a while and allowing those cells to come in. But you can see that we actually do have containment of our bacteria, and then we're going to have more recruitment of more white blood cells so they can ingest the foreign material. So that's going to be, again, cytokines are going to be secreted. They're going to cause the white blood cells that are in the bloodstream to get sticky. And that's sticky, so they're going to have little molecules coming out, right? Those cytokines are going to trigger this, these molecules be put up on the surface of these white blood cells. They're going to stick to the edges of the blood vessel, and that's called margination. Okay, so they're going to stick. And then they're also going to start moving through the endothelial cells of the blood vessel, and that's called diapedesis. Okay, it's a fun word. So diapedesis is squishing through the extrusion of your white blood cells into the connective tissue and where they can undergo phagocytosis and get rid of the infection. So that's going to be leukocyte migration and proliferation. So they're going to grow there as well. Okay, so then they're going to stay there until the infection's cleared up. Okay, so there we go. Continued activity of recruited leukocytes. They're going to phagocytose the infection and until the infection's cleared up. So that is inflammation. The second non-specific defense we have is called interferons. Interferons are specific to viral infections. So these are really cool. You have a set of these interferon proteins that are secreted both by white blood cells as well as virally infected cells. And then those interferons, right? So here, let's follow the picture. So here's our virus infection. The virus comes in and your cell is going to activate interferons. The interferons are going to come out of that cell, and those interferons are going to interact with other cells that have not yet been infected by the virus. And so the interferon is a warning that says to this uninfected cell, there's a virus around, so set up your antivirus defenses. And so this cell, when it binds to interferon, will start to have, the gene expression of antiviral molecules that are ready for that virus infection. So when the virus does infect this other cell, it can basically get rid of the virus immediately. Okay, so really cool interferon. That's what interferons do. Then other non-specific defenses, we have natural killer cells. So natural killer cells are really cool. They work much in the same way that cytotoxic T cells do. And what they do is they can recognize a cell that's virally infected. So here we have a virally infected cell. And when they do have this match of a virally infected cell, they do two things. They're going to put in, number one, they put in these molecules called perforins, and they're going to make a pore, a pore, in the virally infected cell. That is going to then be the gateway for number two, these granzymes. These are enzymes that are going to come in through that perforin, and they're going to be enzymes that's going to trigger apoptosis in the virally infected host cell. So apoptosis, if you're not familiar with that word, it's programmed cell death. Programmed cell death. So your cells have a self-destruct button. It is a series of events that has to take place for the cell to die, but in a very neat way. Cells, if you think about a cell that has a virus inside, and then the cell just dies by breaking open, the virus is going to escape, and it's going to go infect other cells. So we don't want that to happen. So what the apoptotic mechanism does is it creates blebs. So the cell that's on its way to dying, right, is going to bubble. It's going to fracture into little bubbles. So if there was a virus inside of the cell, the virus is going to be trapped in those bubbles, and then that is going to get phagocytosed by your immune cells, by your phagocytes. So apoptosis is a very programmed cell way to die, programmed cell death, and the granzymes are going to trigger that. So it's a nice, neat way to die. Now, if this cell was a bacterial cell, then the perforin can actually just lyse it. So if it's a bacterial cell, it'll just like poke a hole in it and then it'll lyse. So if you look under the natural killer cells in your outline, it can either cause lysis by secreting those perforins, or you can have apoptosis using those granzymes. So that is what natural killer cells do, and also cytotoxic T cells. And then our last couple things here, I think complement. Our last non-specific defense is something called, so before we move on, just another picture I just want to show you. Here's a normal cell, here's our natural killer cell, and we see that we have no killing because what happens is in a normal cell, you're going to have a signal. Don't worry about memorizing the signal, but the signal will say, hey, we're not infected, we're completely normal, and we're going to inhibit the killing response. But if that is not there, right, it's missing, then there's nothing stopping the natural killer cell from killing, so it's going to kill. So it's the default is kill. Okay, complement. So our fourth one is that we have a series of proteins called complement, and you can see that there are many of them, and you're not going to need to know these in any detail, but I want you to know that they do come from the liver. They're in your blood at all times, and they're called complement because they complement the action of antibodies. And what they do is they can do a cool trick. They can create something called a MAC complex, among other things. So they do have other jobs that I'm not really going to make you guys memorize. But the one thing I want you guys to know is they do create something called a MAC, which is a membrane attack complex, which is literally a pore, just like the perforins. So your complement can form a pore and basically stick it into the plasma membrane of a foreign cell and then lyse that foreign cell. So you're disrupting, right? If you're sticking little holes in the bacterial cell, you're disrupting the membrane activity, you're disrupting the water. It's going to, water's going to rush in, it's going to break open. So that's what the complement system can do. So that's a fun trick that they have, the complement system. And I think we have reached the end. So this is what your textbook has. Okay, so this is a bacterial cell, and you can see it. There's a cascade of events. So your complement system is much like how your blood clotting, the coagulation you had like those coagulation cascades where one protein activated another protein, and then that protein, the active form, activates more. It's the same. You have a cascade of events. And then here's our MAC. And so we have fluid rushing in and then lysis, right? So ultimately, MAC complexes always lead to cell lysis. All right, so I think that we are good with this, this first part. Let's go ahead and pause. We'll start the second part.